Good day, everyone. I'm really happy to have you all here. I'm Maurice Choi, and I'm a study director at Cyagen. Today, I would like to share with you about some most recent development on Cyagen's innovative HUGO-GT Humanized mouse models and how they can help you with neuroscience studies. These are my content. Firstly, we will share some general ideas about what is HUGO-GT animal models and their advantages. After that, I would like to share with you about two case studies, two of our HUGO-GT mouse models, how we validated them, and how we characterized them. After that, I would like to give you a brief overview of Cyagen's HUGO-GT mouse models for neuroscience studies. And by the end of my talk, I would like to introduce you about Cyagen platform for neuroscience study. Before getting here, I have been working as a researcher in investigating the underlying mechanism of neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. Like many of my researcher fellows, I use transgenic animals. In fact, to date, transgenic animals remain one of the biggest portion of experimental animals and being employed in discipline beyond the neuroscience, such as human diseases in heart, in lung, in digestion system, you name it. But the thing is, in recent years, more and more researchers actually start realizing that transgenic animals, they are not perfect. Actually, they are quite imperfect in the very job they are designed to do, simulating human diseases. Let me explain why. I would not bore you with the concrete process of how to make a transgenic mouse. I would like to emphasize that the process called transgenesis, where we put human pathogenic genes into a mouse body, though the human gene is specific, the process itself is somewhat random. So we randomly insert a specific human gene into the mouse genome. Since the insertion is random, it may create random disruptions in the approximate locations. In a study conducted by Goodwin et al. In 2019, they investigated 40 strains widely used in all sorts of biological studies in all kinds of human diseases. Out of these 40 transgenic strains, they detected, at least in 75% of them, there is a problem induced by transgenesis in the approximate site. That includes loss of the approximate genes, structural alterations of the gene located approximately, and also, at least, I think, in 50% of those animals, one coding gene is involved. This is what we call unpredictable deficits induced by transgenesis. The randomness of this transgenesis goes beyond just the disruption. It also includes the random location of the inserted genes. As we all understand that every gene's location has its meaning. So if a gene is located in a place where it shouldn't be, that might induce problems. One of that problem is called spatial temporal dislocation of pathophysiology. Let's explain it a little bit. That means if a pathogenic gene is located in a place where it shouldn't be, it may not induce the specific pathology in the right schedule. That means something, if some pathology that happens in human following a specific sequence like 1, 2, 3, 4, 5, in a mouse, if the location is not right, it may not follow the right sequence. Since we all study disease based on its different phases, and we develop a treatment also based on these phases, this kind of spatial temporal dislocation hinders our study strongly. That's what we call unpredictable progressions. In addition to this, the randomness of the inserted gene also includes the random gene dosage. That means even within the same strain, individual mouse may receive different doses of these pathogenic genes. We all know that, at least in most of the cases, the expression level of the gene actually dictates the severity of the specific pathology. This also induces what we call unpredictable severities. So taking all things together, with these unpredictable deficits, unpredictable progressions, and unpredictable severities, transgenic mouse models in the business of simulating human diseases are not as well as we expected they are. Since many researchers have already realized this, they develop different counterparts. Something like they use human iPSC cells, some use human organoid. However, both iPSC cells and organoid are not an entire, fully functioning organism. We, on the other hand, propose a different answer to that. We made a new mouse series. We called it HUGO-GT. HUGO-GT is short for Humanized Genomic Ortholog for gene therapy. The idea of HUGO-GT is simple. We take one mouse endogenous gene and replace it by a full-length corresponding human gene. Since this is a total replacement of a specific mouse gene by a full-length human gene, there would be no random insertion. And this one action actually solves all the three problems we just mentioned. Since this is a total replacement, there would be minimal random disruptions of irrelevant genes. Also, since all inserted human genes are located at the location where their murine counterparts are, we minimize the spatial-temporal dislocations of this pathophysiology. Also, since this is a total replacement, there would be minimal gene dosage diversities. That being said, by using HUGO-GT mouse models to simulating human disease, we could get a controlled pathology, controlled progressions, and controlled severities. However, one should understand that the capability of HUGO-GT mouse models goes beyond this. As I just mentioned, in order to replace the murine endogenous gene, we use a full-length gene, full-length human gene. That means it contains both exons and introns. Of course, introns, they don't end up in functional proteins, but in recent years, researchers started realizing that though they don't end up in proteins, they still play vital roles in pathophysiology of a variety of diseases. The introns, the non-coding genes, they either facilitate or ameliorate the progression of certain diseases. And due to this fact, they are actually quite worth investigating. By introducing the full-length human gene into the mouse, we gave researchers and people in pharmaceutical development a whole new range of therapeutic targets that they could use to investigate the mechanism of pathophysiology of certain diseases, or they could develop new therapeutic strategies from that. In addition to this, the HUGO-GT mouse models could also provide that we called a very accurate study for pathological mutations. Let me explain that a little bit. We generate HUGO-GT mouse line by start generating a wild-type humanized HUGO line. That being said, firstly, we replace the mouse endogenous gene using wild-type human gene. On top of that, we put a specific mutation or specific mutations on that wild-type HUGO line. By then, the wild-type HUGO line will naturally be the best control group for this experimental pathological group. And because there is only one mutation differences between those two things, you could study this specific mutation accurately without being affected by those unpredictable things usually come with a transgenic mouse line. This is what we called the most precise comparisons. There is one thing that becomes more and more important in recent years. That is developing a precise gene or protein targeting therapeutics. So when you use a HUGO line to study a gene-specific mechanism, or you want to develop a gene or protein-specific treatment, you should understand that you are actually studying a human gene inside a mouse body, or you are actually trying to target a human protein, a human gene also inside this HUGO line. That being said, the targeted mechanism you are studying is most likely a cross-species target, a cross-species mechanism, because all the genes, all the proteins are humanized. So by doing so, we could provide with the HUGO mouse line the most translational outcomes. What you see in HUGO mouse models, or what you discovered in HUGO mouse models, will be most likely can be translated to human. Collectively speaking, with all these advantages, the three controlled things, the controlled pathologies, controlled progressions, controlled severities of the simulated human pathology, and some new things, the new therapeutic targets, the most precise comparisons, the most translational outcomes, we believe that the HUGO mouse models are a superior substitute in simulating human diseases in comparison to their predecessors, the transgenic mouse lines. In the field of neuroscience, the very first prevalently used humanized mouse model, I think it should be the humanized A-beta line, developed by Japanese researcher Takaomi Saito in 2014. He replaced in the mouse APP gene the A-beta segment by human A-beta segment with several mutations. And here you can see through time, the A-beta plaques actually form in the brain of these mice. And soon after that, the APP knock-in, or the humanized A-beta, or the APP NLGF line, it has many names, soon become a popular line in both fundamental research and pharmaceutical development. Many of the monoclonal antibody targeting A-beta that we know very well today, the aducanumab, for example, lecanemab, firstly experimented in this mouse. And they all get a very prominent result. What I want to say is, today, in today's research, using humanized mouse models is a new direction. And in accordance to this, we people at Cyagen launched a program, the HUGO-GT program, where we systematically construct and study HUGO-GT mouse models simulating a variety of human diseases. We do this program on two sides. On one hand, we develop a certain HUGO-GT mouse lines simulating human diseases we believe bears significance in human epidemiology. But on the other side, we would like to collaborate with researchers all over the world to construct more, to study more HUGO-GT mouse lines, so we could include more specific mutations, more specific diseases into this category. Because we believe using a more humanized platform, using more humanized mouse models, our understanding to both mechanism of specific diseases or therapeutic strategies for those diseases can be brought to an entire new level. So if you happen to have an interesting project that involves a humanized mouse line, or you would like to create a new HUGO-GT line with us, please do not hesitate to reach out. The contact information for the HUGO-GT program will be provided by the end of my talk. Now that we have some general understanding about the HUGO-GT mouse line, and we have some understanding about why at least we believe, they are superior substitute to transgenic mouse lines. I would like to share with you about two real cases, two case studies about two of our HUGO-GT mouse lines, how we validated and how we characterize them. The first one is the humanized MECP2 mouse line. The MECP2 gene in human is located on the X chromosome, and it's believed to closely associated with a disease we call Rett syndrome. Naturally, this is an X-linked neurodevelopmental disorder, usually affect females. Symptoms of Rett syndrome appear from 1 year old and include, but not limited to, delayed growth, disorders both in movement and coordination, cognitive impairment, and seizures, etc. Here is how we constructed this line. Here you see in the wild-type allele, this orange part indicates the murine, the pink part indicates the human MECP2 gene. By the end of this process, the entire human MECP2, entire murine MECP2 gene has already been replaced by human. Sorry, it seems like my screen share has some problems. Sorry for that. I think now you can see my screen. It is the editing strategy for our humanized MECP2 gene. And on the right side, you can see the gene expression validation. Firstly, using the human MECP2 gene specific primers, we can detect the MECP2 in lungs and brains of those mice only in the humanized MECP2 line. On the contrary, when using a mouse-specific MECP2 primers, we could detect the MECP2 gene only in the black 6 controls. That indicates the successful establishment of the total replacement of the humanized MECP2 gene, in which there is a total replacement of murine MECP2 by human MECP2. Coupled with the humanized MECP2 line, as you may remember, I just mentioned before, we start almost every HUGO line by constructing a wild-type humanized line. So coupled with this wild-type humanized MECP2 line, we constructed a pathological MECP2 line using a most common Rett syndrome-associated missense mutation, which is called the T158M mutation located on exon 4. So we want to validate those two strains in their motor disorders, which is the most prominent disorders in the Rett syndrome. Here is the grouping plan we used in order to do this characterization. You can see we use the black 6 as an active control because we also want to compare our wild-type humanized line to this black 6. We also include the male humanized MECP2 line because though the X-linked neurodevelopmental disorder, Rett syndrome, occurs mostly in females, there are still some records about male Rett syndrome patients. So we would want our study to be as comprehensive as possible. Since male mice have only one X chromosome, there are only two versions of this. One, male mice with wild-type humanized gene, that is, we call it H wild-type, and also with the mutant MECP2 gene, we call it H mutant. And since female mice have two X chromosomes, there were three versions of them with two wild-type. That would be the human wild-type with only one humanized mutant. It's called heterozygote H mutant. And with both HECP2 gene being replaced by mutant human gene, is called homozygote humanized mutant. We plan to test their motor function using two behavior tests. One is called rotor road, another is called gait analysis. Here is the rotor road experiment. The rotor road is just to put the mouse on a rolling road and see how long it can stay there. Here is the latency, which means the staying time of the mouse on the road. As you can see on the male side, it is not difficult to say that the humanized mutant show the most severe reduction of motor function in comparison with wild-type counterparts. In addition to this, the wild-type counterparts behave very similar to the BLAC6 negative control. That means the humanized MECP2 gene, the wild-type one, functions normally in the mouse. On the female side, they behave very similar to the male ones. The homozygous mutant one have the most severe motor disorder, and the humanized wild type behaves similarly to the negative control. What is interesting is the heterozygous humanized mutant behave in between of the humanized wild type and the humanized mutant because it has only one mutant gene. This is actually very similar to what happens in human. With more prominent mutations, human patients with Rett syndrome exert more severe pathologies. Now that we have some data from the rotor road, let's see something from the gait analysis. The gait analysis is a very sensitive method to detect a walking pattern of the mouse. We just put the mouse on the surface and let it walk freely. Underneath the surface, there are detectors to record the walking pattern, the footprint of this mouse. As you can see here, when the mouse walks, the footprint of the forelimb and hind limb on both right side and left side were recorded. And use this data, we can calculate and analyze the stride length and the stride width. If you are interested in this experiment, we can also record the stride sequence, the ground contact duration, pressure, and area. But here, we would only show and analyze the data from the forelimb stride width, hind limb stride width, and also the stride length. Here is the footprint map from both male and female side. The line in between the hind limb trajectory are visualized for an easier visualization of the walking trajectory. The top row indicates the negative control, the BLAC6, and you can see the second row, which is the humanized wild type from male and female, actually pretty much similar to those wild type. That also corroborates that they behave, at least in terms of motor functions, similar to the negative control. The bottom row indicates the humanized mutant from the male side and the homozygous humanized mutant from the female side. And just by looking at them, you already see that they work in a different way in comparison to their wild type counterparts. The heterozygous female humanized mutant is in between the humanized wild type and humanized mutant. However, the difference is not so easy to be visualized just by looking at it. So let's see some statistics. Both on the male side and the female side, let's start from the stride width. It's either a trend or significant differences, but you can already see with more prominent mutant, I mean the humanized mutant on the male side and the homozygous humanized mutant on the female side, you can already see that there is an increase of stride width. On the opposite side, the stride length for both male and female, for both forelimb and hind limb, behave completely opposite. It decreases. It decreases while the stride width increases. And with the most prominent mutation, the mouse exerts the largest stride width, however, the smallest stride length. Just imagine this. The mutant mice actually express such a walking pattern. It has to put its left and right feet as distant as possible and move forward only with a very short step. That is a very typical way of walking without balance, walking with a compromised coordination function. So we put all these parameters in the principal component analysis chart, where we actually analyze multi-dimensional data in two-dimensional chart. And we can see the relationship between those different groups. And as you can see from the left to the right, the wild type ones, the humanized wild type or the negative control, they stick together. And on the very right of this chart, the humanized mutant from the male and the homozygous humanized mutant from the female also stick together. So the wild type one and the most severely diseased one stick to the very left and right side of the chart, and in between is the heterozygous humanized mutant from the female. This distribution actually also echoes to what we see from the rotarod experiments, that heterozygous stay in between the wild type and the mutant ones. So putting all these things together, we can say that the HUGO-GT, the humanized MECP2 line, actually delivered what I just mentioned before. It simulates the Rett syndrome in human, at least on this aspect, the motor disorders, in a way with controlled pathology, controlled progression, and controlled severity. I can give you another example, the humanized MSN2 line. The MSN2 gene is a gene closely associated with another human disease we call spinal muscular atrophy. This is an autosomal recessive neuromuscular disorder, affects mostly infants and children. The symptoms include death of motor neurons, and motor disorders includes also both movement and coordination. But there is a very interesting thing and very different between human and mouse. In human, there are two MSN genes, MSN1 and MSN2. They both make the same protein, MSN protein, but in different proportions. MSN1 makes the majority of this protein, and MSN2 only make roughly 10% of them. But in mouse, there are only one MSN1 gene, which makes 100% MSN protein. So upon MSA in human, the MSN1 is compromised, leave only MSN2 to make MSN protein. That leads to a severe reduction of this MSN protein and leads to the rest of the syndromes. In order to make a mouse line simulating human MSA, there are many ways to make this kind of mouse. Traditionally, we use what we call the triple homozygous line. Firstly, you have to have a MSN knockout line. Then you cross it with another mouse line containing human SMN2 gene, which is the wild type. And when you have this double homozygous line, you cross that with the third one, contains humanized MSN2 gene, which has a mutation, the function is compromised. This triple homozygous mouse line is the most traditional way to make a transgenic mouse line simulate human MSN. It has the slowed growth, and it also has the muscle weakness similar to human, and averagely it has a 70.7 days survival times. What we do here to make a HUGO transgenic line is we just simply replace the mouse MSN1 by the human MSN2. Since the human MSN2 only have a limited ability to make MSN protein, it also induces the MSA similar symptom, includes the slowed growth and muscle weakness. And it has an average 1 month survival time. So with similar symptoms, with an easier way of establishing the line, we actually provide the researchers with a longer time to study the mechanism of this disease and treatment. Here is the validation of the humanized MSN2 mouse model. As you can see here, we can only detect the human MSN in the HUGO line, and we can only detect the mouse MSN in the black 6 control. As in the humanized MECP2 mouse line, this indicates the successful establishment of this line. We also validated the MSN protein in both the black 6 control and humanized MSN2 line. As you can see here, the MSN protein actually expressed a much lower level in the humanized HUGO MSN2 line in comparison to the black 6 control. in the heart, brain, spinal cord, muscle, and kidney. On top of this, we conducted a treatment study using a compound, antisense oligonucleotide similar to one of the most famous drug for the MSA, human MSA, the Spinraza. As you can see here, by using the ASO, what we call the ASO 1027, the humanized MSN line has a prolonged survival pattern in comparison to their counterparts take only the vehicle. And here, as you can see, the pictures on day 33, the untreated humanized MSN2 homozygote express a necrosis on their toes and the truncation of their tails, which is typical for transgenic mouse line or humanized MSN2 line simulating human MSA disease. On the contrary, in the humanized MSN2 mice who received the compound, there is no necrosis and truncation of their tails, even on day 45. Actually, those humanized MSN2 mice can live up to, I think, 190 something days, but definitely more than 120 days. That is several times more than their native lifespan. And we also did a pathology study, as you can see here, using the IHC to stain the motor neurons. The humanized MSN2 mice who received the treatment actually have more motor neurons in their spinal cord in comparison to those who received only vehicles. Here is the statistics. So putting all these things together, this also corroborates what I just mentioned before. Our HUGO-GT mouse line can deliver you the most translational outcomes because the treatment actually works in human, also works here in our HUGO line. The mouse lines I just talked about, the humanized MECP2 line, the humanized MSN line, obviously are not the entire collection of our HUGO-GT mouse models for neuroscience. We also have models for other human diseases such as tauopathy. We have the humanized MAPT series. For ALS, we have the humanized TER-DBP or the humanized FUS. For Alexander disease studies, we have the humanized GFAP. For sodium channelopathy, we have humanized SCN1A, 2A, and 9A. For spinocerebellar ataxia, we have the humanized ataxin, and sometimes we also study the neurological aspects for some muscle diseases, such as the Duchenne muscular dystrophy. So I also put our humanized DMD here. This but a glance of our HUGO-GT mouse model collections, and the list keeps growing. So if you are interested in details of our HUGO-GT collections, do not hesitate to reach out. Well, now that I've talked quite something about our HUGO-GT mouse models, I would like to share with you some information about our CIO platform for neuroscience studies. Cyagen, since its foundation in 2006, has collaborated with more than 7,000 organizations worldwide, and we have contributed in more than 9,000 publications. Some of our collaborators are from the most prestigious companies and institutions and researchers worldwide. We have a high capability of doing animal experiments, and we always hold a very high standard. Here you can see our accreditations. The Cyagen CIO platform can provide you with services from the very beginning of a project, which is the construction of a mouse string, to the very end of it, usually is the pathological examinations. Since I've already talked many things about our ability of constructing mouse lines, I would like to share one or two examples of our other aspects. Let's start with the surgical interventions and drug deliveries. Here we can do, of course, almost all the regular systemic or regional injections, such as the subcutaneous, intravenous, or IP injections, and also the regular stereotactic injections into the central nervous system. What is interesting here is I want to share with some special injections we are capable of. Here is an injection some of you may be familiar with, the convection enhanced delivery. We use a special custom-made device, and this is specialized for delivering large quantity of solutions in the parenchyma of central nervous system. This is demonstration of injecting more than 5 microns of trypan blue into mouse striatum with almost no leakage and damage for surrounding tissues. The only thing you can see is a hint of the needle mark. In addition to this, we can also do intrathecal injection, which is injecting drugs or solutions into the spinal canal without damaging the spinal cord. Here is another example. We injected AV-expressing GFP to the mouse spinal canal at lumbar segment, and after 21 days of inoculation, we examined the GFP expression at cervical segment, which is completely on the other side of the spinal cord. During those 21 days, the mouse behaved completely normally, runs, plays just as usual. Here you can see the positive signal of the GFP, which indicates a successful of the injection. After the surgical interventions and drug deliveries, we can also study the behavior changes, the pathophysiology, using all sorts of behavior tests. Here you can see some of our behavior test method. Besides what I have already mentioned, the rotor road and gait analysis, we can also study cognitive functions using Y-maze, a novel object recognition, a Morse-Water maze. We can study motor functions using the rotor road, here the grip and the treadmill. The social behaviors can be studied by using the 3-chamber sociability test, and the mood can be detected by using the open field, the elevated classmates, or the light dark box. We can also detect pain or sensory functions using the Hargreaves or the Von Frey. Remember, this is just part of the list. And every now and then, we just keep acquiring new methods to our current collections. So with one or several in combination of those behavior tests, you can study different diseases. Almost all those diseases include at least one, usually multiple, aspects of mouse behavior performance. Moreover, we can also study the molecular and pathological aspects of the animals. We can do the examinations at different levels. At genetic levels, we can detect DNA, RNA using the qPCR, ddPCR, NGS. We could also detect protein using usual biochemical methods such as the Western blot or ELISA. What I want to emphasize is our imaging platform, which is capable of doing both in vivo imaging and ex vivo imaging. I could start with the in vivo imaging. Here is one of our in vivo imaging system, and here is an example for bioluminescence detection. The signal you see here is induced by a reaction we call the luciferase reaction. Luciferase is an enzyme which can be excited by luciferin and exerts light at a certain wavelength, which can be detected in a living animal by using this in vivo imaging system. Here, the signal comes from the xenograft, which is a tumor implanted in this mouse containing the luciferase. We injected luciferin, it gets excited and exerts light, which can be detected here. This is a system suitable for detecting substance, implanted tissues, or anything which can take a luciferase with it. In addition to the in vivo imaging system, we could also do ex vivo imaging. And here you can see we have the Laker Thunder imaging system, which is very useful for doing multiplex immunofluorescence staining. Here is a demo. We use the retina tissue and we stain multiple kinds of cells on it. And you can see the multiplex staining result. And here is the brain tissue. You can also see the red signal indicates GFAP, which is the marker of astrocytes. If you are interested in regular IHC or regular stainings, pathological stainings, we have also the regular dark light microscopes. Here is an example of muscle tissue stained by using HE staining. And here is another brain piece with IHC staining, and you can see the staining of microglia. What I want to say is our Cyagen CRO platform for neuroscience study actually is a one-stop platform suitable for multiple neuroscience projects. And we can facilitate you at every step you have along with your project. So if you happen to have a project you would like to implement, or you have an interesting idea you would like to perform, you would like it be carried out, it would be very welcoming that you could discuss that with us, and we would be very happy to help you with that. So we look forward for you to contact us, and we look forward to our future collaborations. Now that I've talked quite something out, I think I can take some questions from the audience. I just saw this question. The mouse technology cost and the turnaround time. Well, the turnaround time is quite difficult to anticipate, since actually it's quite it has to be analyzed case by case. So to do this, we should first discuss with you about the specific pathology you would like to implant and the specific mouse background you would like to use. But what I can say is, on average time, we have a last 6 months record to construct a new HUGO mouse model for any regular uses for pathological studies. For the BAC construct, do we need to send you the BAC construct to construct a new HUGO mouse line? That depends. If the pathology, if the mutations is very specific, you may have to send that. But if it's not, we can also work on the sequence that you can provide with us. Have you systematically evaluated the differences in behavioral tests between HUGO-GT without mutation and corresponding wild type? Well, as I just mentioned along my talk, in one of my examples, the humanized MECP2 line, we compared the wild-type HUGO line with the negative control, the C57 black 6. And that applies to almost all our HUGO-GT mouse line. We always compare the wild-type HUGO line with its background line, which usually is the black 6. And by far, the behavior between those two lines are usually similar, very similar to each other. And I see another question. Is there a specific threshold limit for gene length when developing HUGO-GT humanized mouse? Well, as I understand, for transgenic mouse lines, most of the companies, what they can do is around 100 kilobase length. But we do have a new technology for constructing the HUGO-GT mouse line. With our turbo knock-in technology, we can expand that length to 350 kilobase, so much longer than usual. And I see another question. Constructing mouse models for psychiatric disease is challenging. Does HUGO-GT offers any advantages in this regards? Yes, well, the psychiatric diseases itself is a very, very complicated thing. So what I can say is the HUGO-GT offers advantages in constructing mouse lines simulating human psychiatric disease. What I can say is it's mostly on the genetic level. When you construct a HUGO line simulating human disease, and which is clearly genetically relevant, the gene is come directly from human, and the mutation is also from human. So presumably, at least at the genetic level, the disease manifestation will be more similar in comparison to transgenic mouse lines, which actually contains more unpredictable things. I see another question is, how does Cyagen plan to further expand the application of HUGO-GT mouse in neuroscience research in the future? As I mentioned, by both self-development and collaborating with researchers all over the world, we could construct firstly more HUGO lines, which actually simulates human diseases in a more specific mutations. On the other hand, since these are human mutations, we would like to provide them in more and more pharmaceutical developments. So a specific treatment can be easily developed using our HUGO-GT line. And on top of that, currently you just see I mentioned our humanized MECP2 humanized, MAPT humanized, SMN2. But in the future, we are also considering to construct multi-target humanized HUGO line, which is not simulating just one human gene, but also simulate the entire pathway or entire disease. I see another question. Has Cyagen developed any HUGO-GT mouse models related to Parkinson's disease? If so, are there phenotype validation data or efficacy evaluation data you could share? That is very interesting. Actually, currently we do have a humanized line, which is called humanized SNCA. And we just completed the constructing of this line, and we are in the middle of the phenotype validation. Once we have the data, if you are interested, just contact us, and we are very happy to share those data with you. I see another question. After humanization, how consistent is gene expression compared to mouse genes? Well, that's a good question. Actually, based on what we presume, since this is a total replacement, the random disruption of the proximate gene will be minimized to a level. However, is it completely now? Is it complete zero? We do not know. So we are also planning some sequencing, some total sequencing for some of our HUGO line to see if there is a major change in the HUGO mouse model in comparison to their negative controlled black 6. But what I can say is, based on our current phenotypical validation, there are no clear deviations from the wild-type HUGO line, of the wild-type HUGO line from their negative controlled black 6. So we didn't see some major deficits, which is not related to the gene, to the human gene we introduced. There is another question. If the mutation involves splicing anomalies, how similar are the splicing events between mouse and the humanized model? Currently, if you are referring to the RNA or gene splicing, we assume that is not very, the difference is not very big, very huge between the mouse and human, because in certain HUGO-GT mouse lines, which requires the splicing to generate the pathological deposits such as APP or other things, we didn't see the deviation between the HUGO and the expectation of the human disease they should be simulating. If you are asking for the protein part, it's also, I just take the APP as an example. The APP protein should be spliced by some enzymes and to generate the A-beta deposit. On that aspect, we did see the A-beta deposit in the humanized APP mouse line, but how similar and at which level it simulates the human disease is a question we're currently trying to answer. And also, in addition to this question, as I just mentioned, we are planning to do a multi-target humanized HUGO line. So the protein and enzymes which splice the protein will both be humanized and transferred to the mouse model. I think that would make a much better model. I think that's pretty much all the questions we have here. Well, all of the questions are pretty interesting, and what I can answer here is obviously just an aspect of that. So if you do have more questions, that would be very good for you to just reach out to us, and we are happy to provide you with answers. And as I just mentioned, here are the contact information. If you're interested in our HUGO line, or you are interested in our Cyagen platform for neuroscience, just scan the QR code And there would be the detailed information you can use to contact us. Thank you very much for participating in my talk, and goodbye.